Welcome to Behind the Genes.
One of the great hopes is that some of these new genes that we've found could be useful in preventing cancer. And it doesn't necessarily matter that they're rare, even if they're only 1% of cancers. By using those and changing those in the normal individual before they have had cancer, then we may be able to reduce that risk. So there are lots of potential new targets for prevention that are coming through.
My name is Helen White and I'm the Participant Panel Vice Chair for Cancer at Genomics England. Today, I'm delighted to be joined by Professor Ian Tomlinson, Professor of Cancer Genetics at the University of Oxford, Claire Coughlan, Clinical Lead for Bowel Cancer UK and Consultant Nurse in Colorectal Cancer, and Dr. David Church, a Clinical Scientist Fellow and a Medical Doctor specialising in oncology at Oxford University. Today, we'll be discussing a pioneering colorectal cancer study which, using data from the 100,000 Genomes Project, has uncovered new insights that could transform diagnosis and treatment for patients with bowel cancer. If you enjoyed today's episode, we'd love your support. Please like, share and rate us on wherever you listen to your podcasts. Thank you for joining me today. We're going to be discussing the findings from a landmark study that's been published in Nature. This study used data generously donated by people with bowel cancer who took part in the 100,000 Genomes Project, giving us the most detailed look yet at the genetic makeup of colorectal cancer, better known as bowel cancer. But before we get into that, let's start by hearing from my guests. Could each of you please introduce yourselves?
I'm Ian Tomlinson. I work at the University of Oxford and most of my work is research into bowel cancer, its genetic causes, the genes that are involved in actually causing the cancer to grow, which may be different from genetic causes, and also the use of those data to help patients, whether guiding future treatments or potentially helping to prevent bowel cancer, which would obviously be our optimum strategy to have the biggest impact on the disease and its incidence.
So I'm Claire Coughlan, I'm the Clinical Lead for Bowel Cancer UK, and my remit at the charity is to ensure that everything we do is clinically relevant and that we're providing services that meet the needs of those affected by bowel cancer and the educational needs of those health professionals that work with people affected by bowel cancer. I'm also a nurse consultant in colorectal cancer at Lewisham and Greenwich NHS Trust, and I lead an urgent referral service there and also work with patients with late effects of bowel cancer.
I'm David Church. I'm a medical oncologist and Cancer Research UK advanced clinician scientist at the University of Oxford. I treat bowel cancer clinically and do research on bowel cancer and womb cancer, including a lot of research using samples and data from the Genomics England dataset, which we're discussing today, of course.
Great, thank you. Now let's turn to Claire to learn more about bowel cancer. Claire, can you share with us how common it is, how treatable it is, and if there are any trends in terms of which groups of people are affected?
Of course, bowel cancer is a relatively common cancer. There are about 46,000 people each year in the UK diagnosed with bowel cancer. So that's quite a large number. The thing that really drives us forward in bowel cancer is that the earlier stage you're diagnosed at, the greater chance of survival. So the figures for that are quite stark. We stage bowel cancer through stage 1 to 4, with 1 being the earliest stage and 4 being the most advanced. If you are diagnosed with bowel cancer at stage 1, you have a 9 in 10 chance of being alive and well 5 years after your diagnosis of bowel cancer. And if you're diagnosed at the other end of the spectrum at stage 4, that drops to a 1 in 10. And should people survive after a diagnosis of stage 4, which more people than before do, they will have had a lot of treatment for their bowel cancer. So the burden of their treatment will also be with them after that. So it's really important that we diagnose people at the earliest possible stage, which is why studies such as the one we're going to talk about today are so important. We have noticed that there's been a slight increase in people being diagnosed at a younger age. That said, the latest statistic is 2,600 people diagnosed under the age of 50 in the UK last year. So it's still a disease of older people. You still have a greater chance of getting bowel cancer as you get older. But it's really, really important that we're aware that you can still get bowel cancer as a younger person. Probably one of the most exciting things that's happened for bowel cancer of recent years is our bowel cancer screening programme. And the age for that now has been brought down to 50. We're not quite there all over the country, but in the UK, that's the aim, that everybody will be screened for bowel cancer at the age of 50. So yes, it's a common disease, and staging and early detection is vital.
That's lovely, Claire. Thank you very much for that. David, turning to you, could you please explain to us how bowel cancer typically develops?
Yeah, so we know, compared with many cancer types, quite a lot about how bowel cancer develops because the bowel is accessible to collect samples by a technique called endoscopy, which is putting a camera into the bowel from which you can sample tumours or lumps. And so from genetic research done in the last 10 years, we know that, well, we've known for many years, actually for much longer, that cancer is a genetic disease. It's a disease caused by alterations in genes, and particularly genes that control whether the cells in our bowel grow normally and die normally as they should do. And collectively, when there's alterations in genes that regulate those processes, you can have a cell or collection of cells which are able to grow without restraint and don't die when they should do, which are some of the hallmarks of a cancer. They also acquire the ability to spread elsewhere in the body, which is what kills people with cancer, including bowel cancer. We know from research done in the last 10, 15 years that some of the alterations in genes that can cause bowel cancer in combination occur very early in our life, even in the first, second decade of life, but don't cause cancer. The earliest detectable abnormality is typically a polyp, which is a tumour, a lump within the bowel, which is detectable and, if removed, is almost certainly cured by removal alone. But if it's not detected, then as that grows and acquires more alterations in genes, then it can become a cancer, and cancers develop the ability to invade the bowel wall, to spread to what we call lymph nodes or glands nearby, and also to spread further afield, most commonly to the liver or to the lungs. And for most people in whom bowel cancer has spread to the liver or to the lungs or elsewhere, unfortunately, we're not able to cure their disease, which, as Claire has said, is why there's such an importance in detecting cancers and pre-cancers, as we call them, so that the tumours that are not actually cancerous but come before bowel cancer as early as possible.
Thank you, David. Moving on to the study, Ian, perhaps you can take this. In the study that you carried out, my understanding is that whole genome sequencing was used to investigate the genetic changes that lead to the development and growth of bowel cancer. And for this, participants with bowel cancer in the 100,000 Genomes Project donated both a blood sample and a tumour sample, while those with rare conditions only provided a blood sample. Can you explain why that is?
As you say, the study really looked at two quite separate arms, albeit with a little bit of overlap, as we'll see. So one very important aim was to look at individuals, both children and adults, who had medical problems or other conditions that were unexplained, but which had some features that suggested that they weren't necessarily inherited, but there may be some variation in their genes that had caused them. And roughly half of the programme was dedicated to that. Within that, there was a small number of people who had a strong family history of bowel cancer or who had large numbers of polyps in the bowel, and they were analysed in a way in a separate part of the project from what we're mostly discussing. Within the cancer arm, there was a collection really throughout England of patients who had most of the common types of cancer and a few with less common cancers. And because when we're looking at genetic and related changes in cancers, we need to make sure that those changes have actually occurred in the cancer as it started growing from its earliest stages with a small number of cells in the body that were slightly abnormal and then progressing. We need to look at what genetic variation the patient has in all the cells in the body. We don't want to look at patients and say that looks an interesting change, we may be able to use that if it's present in all of the normal cells in that patient's system. We want to make sure the change is specific to the cancer itself, and therefore we have to sequence both a sample probably taken from blood and a sample taken from the actual cancer. And in a way we subtract out the changes in the blood to identify the changes that have actually occurred in the cancer itself.
That's a very helpful explanation. Does this research show that there's a role for whole genome sequencing in clinical care?
I think my own view is it is all a question of cost. I think that the advantages it provides, it can assess multiple types of genetic change at once. It is relatively consistent across each cancer's genome, between cancers, even between centres, mean that it is the method of choice. There are undoubtedly developments that will happen in the future, maybe being able to sequence longer stretches of DNA at one go that will help the analysis, and some of the computational methods are likely to develop to identify some of the slightly difficult to identify genetic changes. But it ought to be the standard of choice. There are issues and potential difficulties in collecting the high quality samples that have been needed from the pathology laboratory, and that will be difficult going forward with current budgets. And there are lots of challenges, but ultimately it in some form has to be the method of choice. What wasn't done is to look at other molecular tests or assays. Looking at RNA wasn't really done on a big scale, as well as DNA, and other changes to DNA apart from the genetic changes were not looked at. So there are certainly ways it could be improved if you had limitless money, but I think the project 100,000 Genomes has shown that whole genomes they have a lot of advantages and ultimately probably will be adopted by the NHS and similar organisations.
David, could you now tell us about the findings of this pioneering study and what impact these findings might have on people with bowel cancer in the future?
So this is the largest study to date to analyse the entire genome of bowel cancer by some margin. And the fact that we've done whole genome sequencing and in so many people has really given us an unprecedented ability to identify the genetic alterations that drive bowel cancer. And within bowel cancer, we've known for some time it's not a homogeneous entity, that bowel cancer is not all created equal, that there are subgroups of bowel cancers, and we've been able to refine those over previous efforts. And I guess if you were to ask what the biggest take home for me from the study is, it's just the complexity of the disease. So as we've mentioned, we know that cancer is a genetic disease, that it's driven by genetic alterations in genes which regulate the growth of cells or the death of cells or the spread of cells. And we've known for many years that there's a modest number of genes which are commonly malfunctioning in bowel cancer, and they would be in the tens to dozens really. But with this work, we've hugely extended our understanding of the genes that drive bowel cancer. And in fact, we've discovered nearly 250 genes which are altered in bowel cancer and appear to drive the growth of the cancer. Now, we know that not all of those will be validated, and by that I mean that there are associations that we find at the moment, not all of which will be biologically relevant. But interpreting the data, we know that a large number that are previously undiscovered are, or we can be fairly confident of that. And one of the take-homes from that is that many of these are only altered in a small fraction of bowel cancers. So rather than being perhaps half of bowel cancers or a third of bowel cancers, there's a good number of genes, a very substantial number of genes, which are altered in, say, 3, 2, even 1% of bowel cancers. And if we think about how we go about targeting those, and perhaps we'll come on to treatment later. And that poses real challenges for how we work and would think about treating patients with bowel cancer who have those particular alterations in their cancers.
Thank you, David. Yes, we'll come on to treatment shortly, but I think Claire has a question for you.
Yeah, thank you. For me, as somebody who works in this every day, this is such an exciting and interesting study, particularly in light of what we said earlier about early detection and how critically important that is for improving outcomes in people with bowel cancer. So in your view, do you think this research could help shape future screening programs or prevention strategies?
It's a great question. I suppose in terms of screening at the moment, the majority of screening is done in the UK at least by testing for blood in the stool, which is relatively non-specific. So I'm not sure that would be directly impacted by this research. But one area of early cancer detection that's perhaps more relevant is quite a lot of work, including from Oxford actually in recent years, looking at blood tests. So testing blood samples for early detection of cancer, whereby you can test for genetic alterations, fragments of DNA that have alterations from the bowel cancer or any cancer that circulate in the blood. And that tends to rely on a small number of common alterations. And with these data, I could see that we might be able to refine those tests and in so doing improve our early detection of cancer. But that would need quite some work before we could actually say that had real potential, I think. And in terms of prevention, there are, I think, and Ian may want to come in on this, one or two subgroups which you might think that you could try to prevent, but of course that needs a lot of extra work really. But I think we have some clues of the biology of bowel cancer, and particularly some of the subgroups where you might think, well, this drug would work better in terms of preventing that subgroup or that subgroup, but that will need to be the subject of future study.
Ian, did you want to come in on that at all?
So at the moment, prevention is a fairly new way of helping to reduce the number of people with bowel cancer at the level of the whole population, which is what we have in the UK above a certain age group, as we heard from Claire earlier. The methods used, again, as we heard, are screening for occult blood in the stool and then colonoscopy to identify either hopefully early cancers or polyps and remove those. But when we think about the methods that we use for preventing other diseases, then they're normally, where they're successful, using a more easily delivered and, I have to say, less expensive method. So high blood pressure is treated to reduce the risk of cardiovascular disease, and there are other diseases where those, what you might call molecularly based preventive strategies are coming in. We really lack that for bowel cancer in particular. It does happen for some other cancers, but one of the great hopes is that some of these new genes that we've found could be useful in preventing cancer. And it doesn't necessarily matter that they're rare, even if they're only 1% of cancers. By using those and changing those in the normal individual before they have had cancer, then we may be able to reduce that risk. So there are lots of potential new targets for prevention that are coming through. And as David says, it's going to take a lot of work to work out which of those are deliverable and who will benefit. But we have quite a lot of opportunities in that space. And although that may not be us that takes that forward, it may be, but it may not be, we think there's a lot of material for those interested in chemo prevention using drugs of cancer that they can work on and, with luck, deliver some new ways of preventing cancer that may be simply popping a pill every morning to take your risk right down to as close to zero as we can.
Thank you, Ian. David, I think you have something to add here.
Thanks, Helen. One area of prevention which we're really interested in Oxford, and many others are, is using the genetic alterations that we find in bowel cancers and other cancers as targets for vaccination. Now, we know that gene alterations will cause abnormal proteins, which, while they might drive the cancer, make it grow or not die, can also be recognised by the immune system. So the abnormal proteins can be recognised by the immune system as being foreign. And as foreign, they can be targeted by the immune system. So the immune system will try and kill the cells carrying those alterations. And we know for some subsets of bowel cancers, those alterations can be relatively predictable, actually. They occur in quite a sizable fraction of some subgroups of bowel cancers. And one area that we're particularly interested in at the moment and actively pursuing is using those targets. So you need some additional work to demonstrate whether they are particularly recognisable by the immune system, but to use these genetic alterations as potential targets for vaccination with the intention ultimately of preventing bowel cancer in at-risk individuals, or ideally in the fullness of time, the whole population. And we've received some funding from Cancer Research UK to pursue this line of research. And we have a group working on this in Oxford, and as I say, many others do elsewhere.
Thank you, David. Yes, I have a vested interest in this because my understanding is this work is aimed primarily at people with a genetic condition called Lynch syndrome, which predisposes the people who have inherited this gene change or alteration to bowel cancer, womb cancer and other cancer. And I had womb cancer, as I think, David, you know, a few years back, and discovered it was due to Lynch syndrome. And so it's really exciting that you're now looking at vaccinating, preventing, because I take aspirin every day. I have my colonoscopies every 2 years, which have some effect on preventing these cancers, but it's not 100% guaranteed. I don't suppose it ever will be, but having vaccination in that armoury would be fantastic, I think, for future generations. It's very exciting, and we look forward to hearing more about it. Thank you, Ian and David. I mean, we've heard a lot there about preventing bowel cancer, but I think moving back now to potential treatments, we've heard from David how this study has shown a number of actionable findings. But what are the next steps towards treatment? How can these findings be turned into real actions that will benefit those people diagnosed with bowel cancer in the future? Ian, perhaps you'd like to pick up on this to start?
That step is one, in which I'm not personally expert, but a lot of the newer treatments are based on the finding of so-called driver mutations, which are simply genetic changes that occur as the cancer grows and contribute to that growth, and ultimately, if it's not treated, to the spread and dissemination of the cancer. And the fact that we have reported 250, which need validation, but of which a large proportion are likely to be true drivers, means that any one of those can be a potential new target. The criteria to be used for which of those mutations to pursue, which of those driver genes to chase up, are quite complicated normally, depend on many things such as the interests of research groups and small and larger drug companies, and the similarity of those genes to other genes that are involved and the processes that they make to go slightly wrong in the cancer. So there is also the issue that because these are uncommon, everybody talks a lot about personalised medicine or precision medicine. This would be truly precision or personalised medicine because a genetic change that was driving the cancer in only 1% of patients is obviously not a huge number of patients, although bowel cancer is a common cancer, so it's not a tiny number either. But it would mean investment at that level to benefit, let's say, 1 to 2% potentially of all patients with bowel cancer. But I think that's a nettle we have to grasp. And I think our results are showing that most of the really common driver changes are either have not yet been successfully targeted in treatment or are too difficult to target. So we're going to have to start looking at these less common genetic drivers and design strategies, inhibitors, again, that can be delivered to patients relatively straightforwardly in order to see whether they benefit the patients concerned. But there is this problem of getting enough patients enrolled in clinical trials where a change is only present in a relatively small proportion of all the patients with that cancer type.
Thank you, Ian. Presumably, if there's a relatively small number of patients, the people who are looking at running these trials might be looking at perhaps international trials. Would that be one way to go?
So I think David can speak with more personal knowledge, but there are international trial networks and there are collaborations along these lines already underway, I would hope that those could be made use of even more than they are already. There is, a financial consideration for those developing new anti-cancer treatments, which are, high-risk work. And also the costs of setting up trials and enrolling people is not a trivial thing. So I think those are hurdles that can be overcome, but it would need a concerted effort to do that. Patients will play a major role in that, and patient organisations, as well as 100,000 Genomes and other similar projects.
Yes, thank you. David, I don't know if you want to come in on that.
Yeah, the challenge of testing therapies in small groups is a very real one, and there's lots of interest at the moment in exploring alternatives to conventional clinical trials. And as we use more electronic patient records and we have pharmacy records, so there is the potential to get those data from routine clinical practice, and there's lots of investment and attention on that at the moment, so-called real-world data, which is always an interesting term, as if patients in clinical trials aren't in the real world, which of course they are, but it's perhaps a little more cost effective sometimes in clinical trials. Of course, it does pose its own challenges in how you disentangle true treatment effect from other factors, because there are many factors impacting on how long people with cancer live. But there's a lot of investment and effort going into that at the moment. It'll be interesting to see how that develops over the coming years.
Turning to you, Claire, based on your experience, how well do you think people with bowel cancer understand how genomics can help with their care, and what support is currently available to them in this area?
I think the answer, as it so often is it's dependent on individuals and not just one individual. So I think some patients are very motivated to know as much about this as possible and to understand and to know what the next steps may be in their own treatment that may be helped by this. Others don't want to have the same knowledge and want to be guided very much by their medical teams. And I think oncologists obviously are at the forefront of this, and we see at the charity, we have services at the charity that supports patients, and we see lots of queries into our Ask the Nurse service where people have been given variable information about, I suppose, personalised medicine, as Ian alluded to, and how their very specific bowel cancer may be treated. So I think it varies from patient to patient. There is support available, so we have the Ask the Nurse service I alluded to. We have a brilliant patient forum, actually, and everybody in clinical practice will have seen this. Patients often become more expert than anybody, and they share advice, and they're moderated forums that are a very safe place for people to ask questions, where there is a moderator to ensure that it's made really clear that circumstances are individual. And the same with the Ask the Nurse service, because you don't have all the clinical information, so it's about empowering people. So there is support available. I think the other thing that's really important is equipping specialist nurses with the knowledge that they need to support their patients. This is a really exciting area of evolution for bowel cancer, particularly, I think, in all cancers at the moment. But for bowel cancer, I think things have changed fairly rapidly in recent years. And specialist nurses really need support in knowing that they have up-to-date information to give their patients. So that's another challenge for us. And any specialist nurses that might be listening to this podcast, we have online education on genomics for specialist nurses. Just while we're talking about that, and you mentioned Lynch syndrome earlier, so there's been a Lynch syndrome project, as I'm sure you're aware, where we're trying to get testing for Lynch syndrome brought into local hospitals. So there was some funding via NHS England so that the testing be done at time of diagnosis, so a pre-test and then a final test if that's appropriate for everybody diagnosed with bowel cancer to see if they have Lynch syndrome. And in some trusts that has been done, and in others it hasn't yet, and the funding hasn't quite followed in the way that we need it to enable that to happen. It's vitally important. We think there are about 175,000 people in the UK with Lynch syndrome, and we only know about 5% of them. And this is a gene change that is an inherited gene change, so we can do what we call cascade testing, where we test family members, and we can then employ preventative strategies to prevent people from developing bowel cancer. So it's a really important project. So I think as well as supporting patients with the information around the changes that are happening in this area, we also need to ensure that we support the workforce and have investment there to enable the support of all the changes in the genomic landscape.
Absolutely, Claire, and so much resonates there with what you've said. Having myself had cancer, discovered that was due to Lynch syndrome, cascade testing offered to my family members, so valuable. Turns out I inherited my change from my mum, who is 83, has never had cancer, so I think that's a very good example of, it doesn't necessarily mean that you will get cancer, but actually on that point that you made about empowering patients, I always have a wry smile because there's my mum going off to all her other medical appointments, because at 83 she sees quite a few people, and she's always the one telling them about Lynch syndrome and educating them, because most of them haven't heard of it. So yeah, it's really, really important. And that patient forum, you're probably aware of Lynch syndrome UK. I don't have any involvement in that other than being a member, but that is so valuable for people with a particular condition to go somewhere where they can talk to or listen to other people with a similar condition. Really, really valuable. Right. I think circling back really to the 100,000 Genomes project, I think you touched on this earlier, David, but reflecting on what you and Ian have told us about your study, what is it about the 100,000 Genomes project bowel cancer dataset that made this work possible?
There's a few things, one of which, and not least of which, is the sheer size of the effort. So to have whole genome sequencing from more than 2,000 individuals is previously unprecedented. We'll be seeing more of this now as we scale up our research efforts. But at the time of the inception of the project, it was very, very ambitious. And to be able to deliver that is a huge achievement. And the quality and breadth of the analysis is very strong as well. And ultimately, the former is thanks to the people that were kind enough to donate samples to the 100,000 Genomes project. They did so knowing that they almost certainly wouldn't benefit personally from their donation, from their gift, and that any benefits would be some way down the line and hopefully benefit others, which is what we're seeking to realise now. But it's not a given when we treat people in the clinic. So we're very, very grateful to those individuals, and I think also to the scientists who worked incredibly hard over the last 5 years to deliver this work, actually. So having been part of the team, been lucky enough to be part of the team along with Ian, we had hugely motivated individuals that really have dedicated a large fraction of their working lives to delivering this project, which I think was a fantastic achievement as well.
Thank you. Thank you to all those participants who, at a time when their lives probably were turned completely upside down by a cancer diagnosis, were offered the chance to join the 100,000 Genomes project and said yes. As you say, most of them will have known that it won't have helped them, but by donating their data, it's allowed this work to happen, and potentially could change lots of people's lives in the future. So thank you to them.
Could I also just emphasise and agree with it, what David has said? I won't go through all the individuals by name, but if everybody wants to read the published report of the work, there are several people on there. Alex Cornish is their first author, but many colleagues from Institute of Cancer Research, University of Manchester, Birmingham, Leeds, other universities in London that have all contributed, but also colleagues in the NHS and or universities who recruited patients, collected samples, processed them, etc. And of course, the people who did the preparation of the samples in genetics laboratories and actually did the sequencing and basic analysis too. So it is a truly huge effort across particularly all the cancer types, which is particularly a complex collection given the fact the tumour is needed and a blood sample. It's quite difficult in a way to find a formal way of thanking them for all of this, but without them it wouldn't have happened.
On that note, I think we'll wrap up there. A huge thank you to our guests, Professor Ian Tomlinson, Claire Coughlan and Dr. David Church, for an enlightening discussion on the groundbreaking study published in Nature. This research is set to reshape our understanding of colorectal cancer and pave the way for new possibilities in treatment and patient care. If you'd like to hear more like this, please subscribe to Behind the Genes on your favourite podcast app. Thank you for listening. I've been your host, Helen White. This podcast was edited by Bill Griffin at Ventu Digital and produced by Naima Kalachand.